All right, our last cell physiology topic is all about how cells talk to each other. This is a rather involved topic, so you might want to pace yourself and take breaks periodically as we go through this. But in any case, we're going to be learning about how cells send and receive signals. And before we talk about how cells communicate, let's first talk about some reasons why cells might communicate. As backdrop, we need to keep in mind the fact that being a living organism is a very delicate process. And this actually touches on two components of what it means to be a living thing. The fact that living things respond to their environment, and that living things must also practice regulation. So what does it actually mean to practice regulation? To think about what it means, we need to think about the conditions that can be found inside of a cell or an animal compared to the conditions that can be found outside of a cell or animal. In our video on membrane transport, I talked about how the functions of the cell membrane are complex, right? So the membrane keeps certain things out while letting other things in, and it has to keep certain things in while letting other things out. So because membranes can do this, the end result is that the conditions that can be found inside of a cell can be very different from the conditions found outside of a cell. So now if you're just one cell that's bopping around in the environment, what do you do if the environment changes in some way? So for example, here is a teeny tiny prokaryote. This is an E. coli cell right here that's hanging out inside of someone's digestive tract. So let's say that this person now eats a big meal, so suddenly there's a ton of food in the surrounding environment. A cell that has methods that allow it to take advantage of a sudden increase in food will have a big advantage over a cell that doesn't have that kind of flexibility, right? So what we mean when we say practicing regulation is that a cell or an organism is able to detect and then respond to changes in the environment in a controlled fashion. So for the E. coli, this could mean increasing the number of transport proteins that will allow food molecules to be transported into the cell, and then maybe also increasing the amount of enzymes that are present inside of the cell that can break down those food molecules. So now, what if you're an organism that's made out of more than one cell? In that case, you're going to need some kind of method for coordinating your response to the environment. And so what that means is you're probably going to need some kind of more complex method to send and receive signals. So that's why signaling is important. So the next question is, how do cells actually do it? And to start answering this question, we should first think a little bit about the types of things that could be signals in a cellular environment. And there are really two main categories of things that could work as signals. For example, there are physical properties that could serve as signals. So this could be something like a change in a mechanical pressure, a change in voltage, a change in temperature, or some kind of light signal. And note that for almost all these except for the light signal, there'd have to be some kind of direct connection to get these signals from one cell to another. Second, chemicals could also serve as signals. And chemicals have an advantage in that there can be a huge diversity of types to send a wide range of messages. So as we've seen with some of the diversity for certain types of biomolecules. So next, let's talk for a moment about how to organize our understanding about this communication between cells, so cell-to-cell communication. So cell biologists first distinguish between communication that happens locally among neighboring cells versus long-distance communication. So getting a message from your brain to your big toe or from your kidneys to your digestive system. We're going to take a brief look at long-distance communication here, but most of that is going to be a topic for future courses. So long-distance communication usually relies on a combination of chemical and electrical signals. And traditionally, this has been broken down, as I've shown here, into endocrine or hormone signaling, and that relies most directly on just chemical messages, versus signaling through the nervous system, which relies on a combination of electrical signals plus a group of chemical signals. Those chemical signals are called fancy things like neurotransmitters, neuromodulators, and neurohormones. But in general, for the sake of this video, we're mostly going to focus on some concepts that relate to endocrine signaling. So that's long-distance signaling. Local signaling, in comparison, is broken down into three main subcategories based on the details of each mode of communication. And the simplest mode of communication is what happens when there are direct connections between neighboring cells. So in plants, those connections are open fluid channels known as Plasmodesmata or Plasmodesma. And in animals, those direct connections are maintained by proteins called connexin proteins. And in these cases, what this means is that neighboring cells are directly sharing juices with each other. So whatever happens inside of one cell is going to be shared with its neighbors too. So there's really no secrets allowed with gap junctions or plasmodesmata. That's all right, but this can actually also be slow because it's dependent on the process of diffusion. And as we've seen, diffusion across longer distances is very slow. So what else can cells do to get a message somewhere that's more distant more quickly? And that's where a second type of local signaling could come in, called contact-dependent signaling. So diffusion's slow. What else can we do? Contact-dependent signaling. This type of signaling is dependent on a mechanical modality. So the way that contact-dependent signaling works is there's one cell sending the signal that's going to have some kind of molecule on its surface, probably a protein of some sort. And then that molecule is referred to as a ligand. And then another cell might have a different protein on its surface that's a beautiful match for that ligand molecule. So that the second type of molecule is then a receptor molecule. So we've got a ligand binding to a receptor. And as these two neighboring cells kind of move around relative to each other, there's a chance that one of the ligand molecules is going to bump into a receptor molecule and click. So there's a physical connection between those cells. The important thing is things don't stop at the cell membrane. So the signal also needs to travel inside of the receiving cell so it can be detected by that cell and lead to a response. So usually the receptor protein that's on the surface of the cell will be connected to a part of the cytoskeleton inside the cell, and the signal can then be transformed into a mechanical signal inside of that receiving cell. And there's another possibility as well, some form of passage of the signal inside of the cell. We're going to talk a little more about that concept in a moment. For now, just to give you an example of this type of contact-dependent signaling, here's a fluorescent microscope image of a growing neuron or nerve cell, and here's also a transmission electron microscope image of that cell as well. So as these nerve cells grow, they migrate and they have to reach out and find the cells that they're eventually going to connect up with and deliver instructions to. So this is a cell that's looking for a future muscle cell so it can tell that muscle cell to move. Part of the process of reaching out to find that muscle cell involves some contact-dependent signaling. So the nerve cells are literally feeling their way around among other cell types to find those muscle cells to talk to. Okay, so that's the second type of local signaling, contact-dependent signaling. So the interactions between surface molecules of cells that are touching each other. The third type of local signaling occurs by chemical means, where the chemicals are released into the fluid that surrounds the cells, and then the chemicals travel by diffusion to targets that are various distances away. So for this type of signaling, a chemical message will be released, or it's also called secreted by a cell, and then that chemical message is going to float around, and then through the process of diffusion, it may eventually reach or bump into a receptor protein somewhere else. And this type of signaling gets further subdivided according to whether that receptor is present on the original cell that released the signal, so that's shown here, or whether that receptor is on some other nearby cell. So if it's on a cell that's nearby, it's called paracrine signaling. So para means along, so alongside the original cell. If it's on the cell that released it, it's called autocrine signaling. So auto refers to self, so self signaling. And I'm just going to say, for right now, you really don't need to know very much about this autocrine signaling. It generally will show up if you wind up learning more about how cancer works down the road. So instead, we're going to focus the rest of this lecture around processes that are shared in common across both paracrine signaling and endocrine signaling, because both of those processes share a lot in common. The main difference between paracrine signaling and endocrine signaling is just that paracrine signals are local, as shown here, while endocrine signals typically travel to some further away site. So, for example, through the bloodstream of animals, or they could travel in the xylem or phloem in vascular plants. Okay, so coming back now to how hormones work. For any given hormone, what happens is an endocrine gland, a hormone gland, releases a signal and or secretes that signal, you could say, and it secretes it into the blood or the fluid. For the purpose of thinking about this, let's think about a hormone we've already talked about a little bit so far, the hormone insulin. So let's say you have cells in the pancreas that release some insulin into the bloodstream. Then the circulatory system is going to carry that hormone through the bloodstream. And from there, only the cells that have receptors for that hormone are going to respond to it. So not all of the cells in your body have insulin receptors on them. Only some cells do. And so the cells that don't have insulin receptors, they are never going to hear the signal that got released by the pancreas, because they just don't have receptors. And in contrast, cells that do have receptors will receive this signal. To give you another example of this, if you've ever heard of someone having type 2 diabetes, which is also known as adult onset diabetes, that's actually an example of what happens when something changes with the receptor prevalence on cells. So that type 2 diabetes can often be caused by what's called insulin resistance. And what happens, and we don't always know why this happens, but what happens is that cells stop expressing the receptors for insulin. And when that happens, often that makes the pancreas really upset because things just aren't right and those cells are not getting the message. So the pancreas starts to make more and more insulin, just trying to get heard by somebody, and then it burns itself out and it can start to fail. So that's a pretty sad outcome of cells not having receptors to detect a signal. So in any case, just to reiterate this, all cells will be exposed to a hormone, but only the cells that have the correct receptor are going to listen to that message and respond to it in some fashion. And it's such an important concept. Here's another way of saying it. We refer to this as a case of specificity, very much like enzymes and substrates have specificity. So a cell's only going to respond to a signal if that cell has the receptor for that signal. So if it doesn't have the right receptor, it's not going to get the message. Okay, so what happens next after a message reaches a cell? The steps of signaling can be broken down into three main parts. So we've got reception of the signal, what's labeled here as signal transduction, and then we've got the cellular response. And we're going to take a look at each of these aspects in greater detail, and we'll continue to use insulin and a couple of other things as examples. Okay, so again, the process of reception happens when a signaling molecule, also referred to as a ligand, as I said earlier, binds to the protein on the surface of the cell membrane, the receptor protein. So called that because it receives the signal. Then when that happens, a couple of different possible types of changes can take place. First, so since this is like a substrate binding to an enzyme, in general, there will be some sort of shape change that can happen. So for example, here is an electron microscopy image of an insulin receptor that in this case has been stuck into an artificial nanodisc. So when there isn't any insulin present, shown here, the receptor has this particular characteristic shape. But then when insulin is present and it binds, the shape of this whole receptor changes. So it looks really different now, right? And that receptor has now been activated. So one thing I want you to notice here is that in this process, the insulin, so our signaling molecule, is remaining on the outside of the cell. It does not come inside the cell. There can be some other cases where with other types of receptor molecules where different things happen. So here's an example with a sodium ion channel. So you might remember seeing this sodium ion channel before when I talked about facilitated diffusion. And it turns out there are actually some receptor proteins that can also work as an ion channel. So as one example, here's a receptor for a neurotransmitter, so a nerve signaling molecule known as acetylcholine. And when this acetylcholine binds to this acetylcholine receptor, notice the similarity in shape here. What happens is it causes that receptor to change shape, and that opens up the pore in the middle of the receptor, and that allows positive ions to flow into a cell. So in this case, there actually aren't any intermediate steps. The signal changes the receptor, and then that directly triggers a response. There can also be some strange cases where receptors are located inside of a cell. So for this to work, the signal has to be a non-polar substance that can freely cross the plasma membrane. So as one example, a steroid hormone, so estrogen is an example of a steroid hormone, or it's also referred to as estradiol. The steroid hormones, right, they're types of lipids, so they're non-polar, so they have that property. And so these can cross a cell membrane, and then they'll bind to a receptor that's actually found inside of the cell. And these types of signaling molecules tend to have a more long-term response, long-term effect than a molecule that binds on the cell membrane. So again, this is kind of a special case, but you should be aware that there are different types of receptors that can lead to different types of responses. But I want to come back again to that general concept of kind of what happens with a surface receptor, so like what's pictured here. Okay, so that's the reception stage. So for cases such as what I've illustrated here, the next stage for the signals again is going to be transduction. So what that means is the signal gets transformed into some new form inside the cell. So insulin never goes inside the cell, but that signal gets sent inside the cell instead. So the shape change that happens when insulin binds to its receptor then allows for something else to happen inside the cell. And that's what is, this, I know this diagram looks really complicated, but that's what it's trying to illustrate. And we're not going to dwell on any of the details here. You don't need to know the names of the molecules found inside of the cell here. And instead, what I want you to notice here is, so when insulin binds, these other little molecules here, the P dot dot dot, those are all ATP molecules. And what this is trying to show you is when insulin is bound, ATP phosphorylates part of the protein that's found inside of the cell, right? So we've got ATP coming in and then ADP leaving. And there's actually 4 phosphorylations that happen here. Okay, so then once this red subunit is phosphorylated, then it pops free from the receptor, and it bops down here, and it phosphorylates this yellow protein inside the cell. So now this yellow protein has been phosphorylated, and so then it's going to go on, it's going to hook onto this green protein, and the green protein is going to get phosphorylated. And hopefully by now you can start to see what has happened. So the receptor protein is really like just the first molecule in the chain, and it's kind of like a line of dominoes, right? So it's actually very much like a line of dominoes. When you tip over the first domino, that's your initial signal, and then it's that tipping action that carries on rather than the domino itself. So that is signal transduction. And just like setting up a bunch of dominoes, there are reasons why cells use this process of signal transduction. So for one thing, signal transduction is very fast. So think about what would be involved if you needed to get a piece of information across a big chunk of the US. And your options were to either have that information hand-carried in a letter by the Pony Express or transmitted in a telephone call or a text message. So in this case, signal transduction would come to the rescue. So much, much faster. Second, signal transduction can be really versatile. So for example, at the transduction step, it's possible for one signal to kick off a whole series of events inside of the cell. This whole process is referred to as a cascade. Or that signal could get amplified. And to illustrate this, so that signal amplification, it's very much like what you see here. Or if you knock over just one domino, you can have a domino effect and knock over an exponential number of dominoes. So that's signal amplification. So signal transduction can be very versatile. Okay, and this is actually true for our insulin example also. So the cartoon here shows an overview of this in a slightly different way. So here's our insulin binding to the insulin receptor, and that insulin signal actually does two different things in a cell. So it sets off a whole series of events illustrated here. So here's a cascade that generates a growth signal, so tells a cell it should grow. And it also sets off a series of events illustrated over here on the right. So again, a signaling cascade that happens that favors the uptake of glucose by causing the installation of some glucose-facilitated transport proteins on the outer cell membrane. We'll see a little bit more of that in a bit. Okay, so signal transduction is versatile. So our third benefit for signal transduction is one where actually the domino analogy kind of fails. And the third benefit is that the signal transduction process is easy to turn off. And I think the best way to illustrate this is to look at two different examples. So first, here we are looking at events at the tip of a neuron that releases that nerve signaling molecule I mentioned earlier, acetylcholine. So when acetylcholine is released, it diffuses across this small liquidy space between the cells, and then it binds to receptors on a neighboring cell. But there's another thing lurking in the space here between the cells, and that's an enzyme pictured down here called acetylcholinesterase. And what this acetylcholinesterase does is it specializes in breaking down acetylcholine. So after some time has passed, the acetylcholinesterase will break down all of the acetylcholine that's in this space, and at that point it will have shut off the signal from this upper neuron to the lower neuron. Okay, so signal transduction can be turned off. But now let's look at our estrogen example again. So here the receptor is inside of the cell, and it's kind of an involved journey for this estrogen to reach its receptor, right? So by the time it gets there, it's going to be all over the place, like all over inside the cell, all over inside of all other cells, just kind of like a party guest that won't leave. So it takes a lot longer for a signal such as estrogen to get turned off. Okay, so that brings us through signal transduction. So might now be wondering, well, what about this whole response step? We're going to dive into aspects of the response step more down the road, but for right now, we're just going to look at our examples again to get some ideas about what a cell's response could look like. And again, don't worry too much about the details. These little cartoon items all have names, but we really don't, the names sound like complete gibberish, so we really don't need to worry about what they're called. The main thing we want to notice is that eventually, through this whole process, the word makes its way over to a vesicle, so illustrated here, and this vesicle has some of these, what are illustrated as GLUT4 glucose transport proteins in it. And this whole process with insulin signaling causes this vesicle with GLUT4 transport proteins in it to migrate to the cell membrane and then fuse with it. And so now there are GLUT4 glucose transport proteins in the cell membrane. And now there's a route for glucose out here to get inside of the cell. So that's one type of response. For our estrogen example, the response is a little different. So this estrogen and receptor are going to bind to substances that are inside of the cell's nucleus, and they're going to turn on genes that hadn't been turned on previously. And those genes basically are going to cause the cells to grow. Or if you really wanted to get more technical about it, those genes are going to cause cells to proliferate, so to increase in number, to differentiate, so to turn into more specialized types of cells, and also to survive for longer. So that's another type of cell response. Okay, so I have one more example to share with you for cell signaling, and then I'm going to tell you about a clever story. But I'm also going to note this is probably a good point to pause for a couple minutes and take a break, because we've already covered a lot of ground in this video. So consider pausing, take a break, and then come back for these last two items. All right, so here's another signaling example. The insulin receptor that I showed you earlier is one type of receptor that's found in cell membranes. And this other doodad that's illustrated down here is a second type of receptor that's extremely common in cell membranes. And this second type is a type labeled here as a G protein coupled receptor. Now, as I note here, there are hundreds of different kinds of these G protein coupled receptors. It's a very common receptor type. So it's kind of useful to just get a sense of how they work in general. So what happens is a signaling molecule, such as a hormone, pictured here, will bind to this receptor and change its shape. And so, an example could be the hormone epinephrine, also known as adrenaline, binding to a receptor in the liver. That's epinephrine binds to a G protein coupled receptor. So that change makes this G protein coupled receptor very attractive to a protein illustrated here that's found on the inner part of the cell membrane. So this is the G protein. And it's called that because it can bind to an energy transferring molecule known as guanine triphosphate, or GTP. And GTP is very similar to ATP. So hormone binds, G protein coupled receptor goes, all right, get over here, G protein, and bring your friend GTP over here also. Okay, so now that they've come over, so once GTP has bound to the G protein's then activated, and it heads over to its friend in green here, adenylate cyclase. And then this G protein is all, okay, adenylate cyclase, I hear that you really like to boogie with ATP. So then what adenylate cyclase does is it's going to convert an ATP molecule into another substance, and not too worried about names right now, but the substance called cyclic AMP. And cyclic AMP is then the new messenger inside the cell that continues on, and it's going to wake up other enzymes and make those other enzymes get to work. So for example, here's one called protein kinase. And protein kinase is a workhorse that sticks phosphate groups on other enzymes, phosphorylates them, and makes them get to work. So that's our cellular response here at the end. Okay, so hopefully you can see here there are a couple molecules involved in this whole process of signal transduction before we finally get to the cellular response. So now we have just one more question to consider, which is a question of, well, how on earth did people ever figure out this was how cell signaling worked? So specifically, how do we know that there have to be multiple relay molecules involved instead of just a single molecule that just does its thing? So this is really figured out by an American guy who was named Earl Sutherland. And he was really interested in how glycogen, a carbohydrate, can get broken down in the liver into glucose molecules. And what he did is he would add the hormone epinephrine to a bunch of liver cells, and then when he analyzed the results, he'd find that a bunch of glucose got created. Great. And along with that, he also figured out there's a specific type of enzyme present in liver cells. It's called this named glycogen phosphorylase that's responsible for glycogen getting broken down into glucose. Okay, so that's great. So, but here's the kicker. You had a question. So, based on this information, if you add epinephrine to get glucose, and the enzyme that does this is glycogen phosphorylase, then if you mix these three things together, what should happen? Automatically, you might think you'd expect to get glucose, right? Because you got epinephrine, glycogen phosphorylase, and glycogen, so you should get glucose. Except that's not what happened. When he mixed those things together, nothing happened. So, based on this outcome, Sutherland concluded that there must be at least one more step in the middle that's missing from this process. And so eventually he figured out that the liver cell membrane had to be intact to make this signal transduction process take place, to activate the glycogen phosphorylase and actually get glucose breakdown. So that's pretty amazing. And you can see this again on a video link that's provided with the lecture slides. All right, so having said all that, let's bring ourselves back to this bigger picture again. So in this video, I've talked about why cells communicate, but mostly about how cells communicate. We've seen several different examples of different types of cell communication. So ranging from some long distance communication by hormones to extremely close range communication through gap junctions, where cells are literally sharing juices with each other. And we've talked in exhaustive, maybe exhausting detail about just how chemical communication can take place. And that's through that three-step process of signal reception, transduction, and cell response. And I want to emphasize here that if you go on to study cell biology, you will learn a whole lot more about specific examples of these specific types of signals. And so for right now, what I really want you to do is just mostly focus on the big picture as illustrated by the handful of examples that I brought in. So becoming familiar with these examples should help you to see how it is that cells are able to respond fairly quickly to any changes that take place in the environment. And so these processes are critical for helping to keep cells alive through all of the ups and downs that can take place in the cell's environment.